Cloning of cattle has been a focal point for many scientific processes, such as the experiment conducted by Dr. B. Ovach and D.N. Wells. A popular method of cloning is somatic cell nuclear transfer, or NT, occurring in over 20 countries and 130 laboratories. 75% of these work with production animals, and of this, 75% are focused on the cloning of cattle. While cloning could be deemed as a productive venture both financially and biologically, it is yet to be perfected to where there are no fetal casualties. Zona-free NT is conducted in this experiment, testing its effectiveness based on fatality rate. A number of chemicals were needed in order to carry out the ultimate procedure. These include DMEM F1210, HDMEM F1210, penicillin streptomycin, fungazone, M199, B199, H199, H19910, B19910, IVM medium, H199 PVA, and HSOF. Other materials required are a source of oocytes as well as fibroblasts, which are collagen productive connective tissues from both adult and fetal sources. The steps taken are as follows. Firstly, isolation of the adult ear skin fibroblasts occurs, followed by the isolation of the fetal fibroblasts. Next, the G1 donor cells are mitotically selected and prepared. These processes succeed in collecting DNA from both donor cells in order to later transfer to oocytes. Succeeding this, oocytes are located from mature slaughterhouse ovaries.
As can be seen and described in this diagram, enucleation is the next step to occur and where all previous DNA from the oocytes is removed.
Cell attachment is the first step of NT, which is followed by cell fusion. Both are processes of inserting donor cell DNA into the oocytes. The cell is then artificially activated by a chemical stimulation, creating reconstructed embryos in vitro. Succeeding this, the embryo is cultured for a total of 7 days. The final process to be undertaken is the transfer of the embryo to the surrogate parent and the subsequent preservation of this embryo within the gestation period. Although natural carving is encouraged, if major complications occur, then a caesarean is performed, although this occurs in less than 10% of all cases. The whole experiment was repeated in a total of 194 surrogate parents.
The entire process is diagrammatically shown in this figure. The results taken from this complex procedure can be seen in this table.
Testing that was undertaken found that only 47% of the 194 embryos actually resulted in a pregnancy. The number of the total that went on to full term was even less, just 27 out of the 194. A further 10 offspring were unable to reach the weaning stage, which leaves a 9% success rate. These results further emphasise the need for further research into different cloning techniques in order to ensure a higher success rate of offspring via cloning methods.